Hello there guys, Teacher Krishna here, and welcome to our next class for the third unit. Of course, this is a class that is part of VHAV School, and of course is intended to 9th high school. Just like you know, our subject is biology, and we are working on week number 23 today. Let's go over our presentation. Remember to have your notebook with you in order to make today's summary. And of course, make sure that it includes the following parts: date, end in mind, title, summary, and some extra decoration in general. All right. Here we have our topic for today, 3.07, biotechnology. And the end in mind for today is the following: explain how technology helps to compare and study genes and proteins. All right, so make sure to keep track of the end in mind in your notebook. I'm going to give you a small summary of activities that you need to do today. First of all, watch this YouTube video and make a summary in your notebook. Number 2, read topic 3.07 and complement your summary. I suggest you especially focus on pages 4 and 5. And lastly, study your summary, then complete the assessment for topic 3.07. Pretty simple activities for today. All right, let's continue. And let's start with biotechnology, and we have a glowing cat right now. Very quick observation. Do you think it's possible to have a glowing cat in real life? Yes or no? Well, let's find out. In 2007, scientists in South Korea modified the DNA of a cat. So when a certain protein was produced, it will glow red in its skin after exposing it to ultraviolet radiation. So the answer will be yes, it is possible to have a real glowing cat in, of course, real life. It's something pretty interesting, but it's part of the topic biotechnology. So by modifying a little bit the DNA of a species like a cat, we can generate total different reaction in its body. This was a great step into the world of biotechnology. Right, let's continue talking about genetic engineering. And basically the process that you see right here on the left on that picture is a process that scientists use in order to modify the genetical blueprint of an organism. So genetical engineers, they analyze and change the genetical makeup of a living cells. In some cases, they add genes from one organism to another. It is possible because all living organisms are shared, or most likely share the same universal code, which is DNA. All right, so how can we relate this to the picture we have right here? So this is the DNA from an organism, and this is the DNA from another organism. So people cut it right through, then they actually stick it together, and the resulting genetical material is actually a DNA that has different information than the original one. A recombinant DNA. So DNA that is artificially formed by combining DNA from different organisms. So basically what you can see right over here is recombinant DNA, mixing the DNA from two different organisms. And these are the steps that scientists follow in order to make this process possible. So first of all, extract the DNA sequence that contains the gene that wants to be inserted. Like in this case, let's suppose that this is the part that wants to be inserted. And number 2, restriction enzymes cut DNA sequence at a point where the nitrogenous bases can pair up. All right, so here we have the restriction enzyme, and it is going to cut DNA on both sides. And lastly, number 3, the DNA sequence will be sticked together thanks to the DNA ligase. Okay, and basically right over here we have DNA ligase, and just like you can notice, the nitrogenous bases are pairing up again. All right. Oh, and I'm sorry, it's not ligase, it's ligase or something like that. I need to check on that word right now because I'm doubting about it. But it remind me about that specific word during the meet session so we can take a look at the pronunciation. It's ligase from what I remember. I'm sorry about it. Okay, now let's talk about transgenic organisms. So transgenic organisms are basically organisms that have been genetically modified. So for example, right over here, we have the original organism, and here we have a transgenic organism. So why people do this kind of stuff? Let's go over some information and then we can go back to this picture. So transgenic organisms are organisms with recombinant DNA. And this type of technology was refined in 1980s with the use of mice. So just like you can notice right here, we have several mice, and scientists actually try to change their DNA in order to, of course, generate some certain results that they were looking for. Now it is possible to produce transgenic plants, animals, and microorganisms. They are used to produce improvements in agriculture. So most of the time, people are going to use these transgenic organisms in order to have like a better income, or to have like better organisms to commercialize with. So here we have several examples. So we have the Bt toxin, which is a type of protein that is harmful for insects but harmless to humans and animals. So what happens is that farmers have noticed that there are a lot of insects that are going to affect their plantations. So in order to change that, what they have done is that they have actually modified the plants a little bit so they could actually produce a Bt toxin, which is going to cause that when an insect is trying to eat some of the plantation that they have, those insects are going to die. But it's very interesting because it only affects insects, but animals and humans can consume this Bt toxin without any inconvenience. Or so they say, right? It's always good to have a very interesting doubt right there. We have manufacturing. Engineers are using transgenic plants to have a better result when producing plastic. Of course, probably what most people want to do with this kind of organisms is to produce more. That's the main goal, right? Because the more they produce in the least space, of course, the more earnings they are going to have later on. We have golden rice, which is transgenic rice with higher amounts of beta-carotene. When humans or other animals lack this specific nutrient, which is beta-carotene, it is going to cause several illnesses in the organism, of course. So actually, this golden rice is very beneficial for humans and other organisms, because when they consume it, they are going to have higher nutrients in their organism, which is going to cause a better health. So this is one of the main goals that transgenic organisms have, like, of course, from a scientific point of view. The scientists want to produce like this kind of food that people can consume in order to have a better health. And lastly, we have transgenic animal. Cows are injected with hormones to produce more milk. Of course, we're talking about the United States, where most of the milk that they consume come from cows that have been artificially treated with hormones. But there are some other organisms that are actually treated with this kind of treatment. So what happens is that, of course, farmers are going to want the cows that have way more graze, or that they have way more muscle, in order to produce more meat out of one cow. So it is possible to modify, just like in the example of the mice that we have right here. So they were modified in order to be bigger. So the same thing can happen with cows. They can be artificially modified in order to be bigger, larger, and to produce more products for the human consumption. Now we have another very interesting thing, which is cloning. So cloning, it means using a single cell from a grown-up organism to grow an entire identical organism. Unicellular organisms are easy to clone, not like pluricellular organisms. And the first successful clone was done in 1952 with amphibian tadpoles, which is basically a type of frog at a very early stage. And another great success was in 1997 with a sheep named Dolly, who unfortunately had to be put to sleep in 2003 due to a lungs disease that is common in much older sheep organisms. So it was a great advance in science because actually I have a picture right over here. So the little sheep that you have right over here is actually a clone or basically an identical organism as the grown-up sheep. So it's very interesting to analyze that something like that can actually happen. But the problem was that it developed a disease in its lungs that is only typical of much older sheep, which also caused a great discussion in the scientific community. Animal cloning. It uses a process called nuclear transplant. The nucleus of an unfertilized egg is removed and replaced by the nucleus of a diploid body cell. It generates an embryo that will grow an identical organism. So basically, we have an unfertilized egg. So then we are going to remove one cell from a grown-up organism, just like this sheep. We are going to extract the nucleus from the cell that we previously extracted from the grown-up organism, and then we are going to insert that in the unfertilized egg. So basically, when it grows and it generates another organism, it's going to be an identical, genetically identical organism to the donor. All right, let's go to DNA fingerprint. And this is something very interesting as well if you want to study criminology later on. Cells from tissues samples are used to extract DNA information and through the restriction enzyme section it. We can use several cells in order to analyze the genetical material such as sperms, blood, saliva, and much more. The DNA segments are separated according to their length. Basically, this is called a gel electrophoresis. And a radioactive substance called DNA probe is added. It combines with the gel electrophoresis to produce a fluorescent image. It is used to get the fingerprint of the DNA and compare it to other samples. And of course, it is used in forensic science. So what happens when, for example, we have the scene of a murder like right here? But actually, the police officers found a gun that was dropped on the ground, as well as some other samples of blood. So how can we actually find who the criminal is? How can we find the person that committed this act right here? Well, of course, we can use DNA from, for example, the gun. And that is why the police officers and the personnel that actually take samples from the crime scene are very careful with everything they do. Just like you can notice, like they are using masks, they are using a protective suit as well as gloves. So their genetical material actually is not on the samples that they are taking. Because imagine if probably the sample of the saliva of the investigator goes to the gun, then of course he can be accused of committing the crime. So what the investigators do is that they try to find a compatible DNA information from the gun or from the blood from the crime scene. So of course, since the DNA from each person has this, let's say, a unique pattern, they can actually trace him or her down. Right, and that's basically it for today's session, guys. Thank you a lot for coming one more time. And remember to go back to the activities section so you can remember the activities that you need to complete today. Any questions, I'm available on WhatsApp, and I will see you during the live session just in a while. So goodbye, guys, and take care.